The overall goal of this procedure is to conduct an immunofluorescent stain for active zone proteins in Drosophila third instar larvae. This is accomplished by first selecting and pinning the larvae down onto the dissection dish. The second step of the procedure is to dissect the larvae, removing excess tissue to expose the muscles and neuromuscular junctions. The third step of the procedure is to immunostain the dissected larval pelts directly in the dissection dish. The final step of the procedure is to mount the larval pelts on a microscope slide. Ultimately, results can be obtained that show neuromuscular junction active zone morphology through immunofluorescence microscopy.
This method can help answer key questions in the neurobiology field, such as regulation of neuromuscular junction proteins, like ones involved in active zone morphology.
To create a dissecting surface, pour Silgard 184 silicone elastomer base into a small tissue culture plate. Make sure not to fill the plate completely so that the dissection area is lower than the rim. Trim dissection pins to a length of approximately 3 to 5 millimeters for increased ease of manipulation, and make sure to have at least 6 pins per larva. From wild-type and mutant strains, identify and select wandering third instar larvae that are actively crawling around the wall of the vial and have not begun to pupate. Remove 2 larvae from each genotype from the vials and place them in a washing dish filled with PBS to remove debris. Under a stereo microscope, wick away excess moisture on the dissecting surface using a Kim wipe. Arrange the larvae so that the dorsal side is facing up and the 2 white trachea are visible along the dorsal side of the larvae. Center the tracheas in the middle of the larva as they mark its dorsal midline.